All right, thank you so much for that introduction, and to the organizers for the opportunity to be here today, learning from all of you, as well as sharing some of the work from what we've done at CZI. So today I'm going to be talking about applications of CZID, which is a free metagenomic platform for pathogen surveillance, and really focusing on a pilot study that we did looking at causes of child health and mortality. So just by way of a little bit of background, CZI's infectious disease work is really focused on accelerating the detection, identification, and tracking of infectious diseases. And so we do this by partnering very closely with the Zuckerberg Biohub in San Francisco to provide technology tools as well as training needed to enable high-resolution detection of any pathogen around the world. So CZID is a software platform that's emerged out of this collaboration between CZI and the Biohub. And it's an open source cloud-based metagenomics platform really meant for researchers. And so what this platform enables is anyone with a laptop and an internet connection to do metagenomic data analysis, which is traditionally pretty difficult to do, might be very compute intensive, and so this removes a lot of those barriers. And the way it does that is allowing for a simple data upload and cloud-based data processing, data management for samples and projects. It generates single sample reports of microbial and AMR abundances, and then allows multiple sample analysis and flexible downloads for offline analysis. So altogether, CZID serves really a wide range of different use cases. Since it can accept RNA and DNA sequencing data from any sample type or host organism, there are researchers using it for kind of all different questions. So some folks are studying different causes of mystery illness. When standard diagnostics fail to identify something, metagenomics often can. They're also interested in emerging biological threats, so things like AMR or novel virus discovery. Some folks are zooming out and looking at the microbial landscape across a cohort of many samples, and others are zooming in on one particular pathogen to understand if there's a particular disease outbreak happening. So underlying the platform, there are a number of different workflows. The ones we'll be talking about today, we have metagenomics, where the goal is to understand what microbes are present by matching sequences to databases of known organisms. The consensus genome pipeline, which provides the input to downstream genomic epi, which creates those consensus sequences from raw sequencing data. And then AMR, which is really the goal there is to understand the presence of AMR genes, and this relies heavily on the CARD and RGI tool suites. So the workflow code available for these is available at GitHub, as shown on the bottom. But I really won't be talking too much about the details of those pipelines today. I'm happy to chat afterwards if that's of interest, but I really wanted to highlight this pilot study that was done with the CHAMPS network. And so CHAMPS stands for Child Health and Mortality Prevention Surveillance. And the goal of this organization is to collect, analyze, and share evidence to really prevent child mortality in regions where it's highest. So for this part of the talk, I'm going to be going over a little bit of background and experimental setup that went into this study, and then providing a case study highlighting the way CZID was used for one sample in the project, and then zooming out to give some learnings from a cohort-wide study, really with the aim of evaluating how is metagenomics potentially augmenting the existing diagnostics that are used as part of that network. So this pilot study involved 81 subjects. They really represented a range of different ages, as shown here, as well as infectious and non-infectious causes of death. And so for each of those subjects, we had multiple sample types. And so ultimately, there was paired RNA and DNA-seq done from 233 samples. So those were lung, CSF, blood, and some water controls. So those raw FASTQ files were then uploaded to CZID, and the taxonomic abundance results were analyzed, as I'll show in the next few slides. So data, I'm showing here the data once we uploaded the project. This can be browsed in the CZID web portal. And I should clarify that only the uploader and their specified collaborators have access to this until they make the study public. But diving into this particular case study, so this was a patient with a pneumocystis jirovecii co-infection. So the CHAMPS network did their standard battery of tests and identified pneumocystis jirovecii, streptococcus pneumonia, and cytomegalovirus in the sample. The initial cause of death was listed as respiratory infection, but the underlying cause of death was later identified to be HIV. So when we look at the sample report in CZID, we see that, lo and behold, Pneumocystis jirovecii is the most abundant organism. So this is really great for confirming the presence of this organism. We can then use the coverage visualization to really see that we had high coverage of that organism across the genome. We don't want to go through every sample one microbe at a time. So here we took all the samples associated with that patient and generated this heat map that allowed us to visualize what organisms were present across the samples. So here, kind of the metadata is across the top, where DNA is in turquoise and RNA in purple. And you have your samples from left to right, lung, CSF, and then blood. And so right away, some things that we notice here are that the RNA sample seems to have more pathogens in it. Or the lung samples have the most pathogens, and RNA even more than DNA. And within that, we start to notice that there is an HIV hit showing up in that RNA sample from the lung. So we can go back to that sample report and see that, yes, we're finding this at significantly lower abundance than the other organisms in that sample, but still somewhat significant coverage here. So we can then use the CZID's consensus genome pipeline to generate a consensus sequence. Ideally, we'd be then able to take this into downstream genomic epidemiology, but here we see that we actually only got 30% of the genome, so that won't do for this particular case, but could give an example of how that might be useful. So altogether, kind of wrapping up this case study, we saw in the metagenomic data that there were a couple of true positives as compared to the standard clinical testing done by the CHAMPS network. So in particular, 2 of the 3 organisms were detected, the pneumocystis as well as the CMV. There was, however, 1 false negative. So this was the Streptococcus pneumonia was not identified by metagenomics. We have seen that in some cases where some PCR-based diagnostics often can be more sensitive than metagenomics if you really do know what you're looking for. But we also see that metagenomics was able to identify the HIV, which was not identified by the standard battery of tests, likely due to lack of suspicion at the time of collection of that sample. So highlighting the potential for increased sensitivity given the way that samples are processed. So we've shown here how the single and multi-sample analysis tools could enable the identification of these microbes. But that was just for 1 sample. And so we wanted to zoom out and do this across the cohort. But we could have done this in the web app, but being a bioinformatician, I wanted to kind of download that data and make it a little bit faster for myself. So I took those sample reports out of CZID and then grouped them by patient. So now you're seeing kind of the conversion to data visualizations that I'll be using through the rest of the talk, where we're focused on a set of pathogens. And in particular, we wanted to, one important thing with metagenomic data is making sure that we're not just finding contaminants. So that was mentioned earlier today. So CZID provides metrics of abundance as well as significance with respect to the water controls. And so here what we did is we looked at, in blue, there will be things that are both abundant and significant as compared to the water. And then in green are the low abundance things, but they're still not present in the water controls, so likely potentially interesting pathogens. And finally, in purple, we have the abundant but insignificant. So they might be highly abundant, but maybe there's E. coli in the water, and so we can't really trust that finding. So also the X in these heat maps denotes pathogens that are outliers within the sample. And if there's an outlier that's also a known pathogen, there's been some work to show that that often corresponds with some clinical relevance. So from here, we bin the data into groups based on likely infection, where we had lots of high abundance outlier organisms that are known pathogens. Possible infection, where you had maybe a smaller number, not across all the samples, maybe lower abundance, but still significant. And then unlikely infection, where there wasn't really anything found by metagenomics. We could then take those results and compare those against the orthogonal test results run by the CHAMPS network. And what we found was that we got 13 true positives, so things that were concordant between metagenomics as well as the standard diagnostics, 27 that were concordant true negatives, and then 9 cases of missed known pathogens. And so as I mentioned before, that can happen given some of the ability to amplify things using kind of PCR-based approaches. But we did notice that the majority of missed cases were CMV. And so that's actually something we've seen in another study as well. Seems like metagenomics has a little bit of a hard time with that one. I can't quite explain it. But importantly, on the upper right, we see 27 potential new pathogen hits. So these are cases where the standard diagnostics didn't identify something, but they might be of interest to us. So things like Klebsiella pneumonia and a range of other gram-negative bacteria. There are also a number of viral hits. So we can look across the cohort. And here we're showing the RNA viruses that were identified, most of them at relatively low levels. But from some of these, those with the star, we generated pretty decent consensus genomes that could, in theory, be taken downstream into further analysis. Another observation that you may have noticed in some of the slides, but worth calling out specifically, is that we've observed that in this pilot study, RNA tends to be more sensitive than DNA. You'll see that the RNA, which is the ones with purple headers, versus the turquoise is the DNA. And the RNA tends to have more organisms identified, as well as higher abundance of the organisms that are present on both. And so this is consistent with some results that have been shown previously. I highlight here the Zinter et al. Paper, but interesting as far as study design if we're thinking about implementing this. And finally, this was kind of a really cursory look, but running the AMR pipeline, what we see is that there are AMR genes present across several of these samples. This would require a lot more work to dig into and kind of really understand what's going here. But it's interesting that metagenomics allows us to capture both of those pieces of information and has potentially valuable use downstream. So summarizing the findings of this cohort-wide analysis, we saw that RNA-seq tended to be more sensitive than DNA-seq. In this particular cohort, the lung samples tended to have more likely pathogens. That could be just based on the distribution of samples collected, but also the lung having more organisms generally. Metagenomics was able to pick up 100% of the known Klebsiella pneumonia cases, but it did really struggle to pick up the CMV cases. There were a number of potentially new pathogen hits, and the AMR genes that we saw present in the samples are interesting and might be something to look in further. So altogether, metagenomics provides a valuable method for identifying known and potentially unexpected pathogens. So analysis of this data can typically be relatively complex, but can be simplified through the use of tools such as CZID. We suggest that future studies might consider adding in a component of metagenomics, if possible, to do some of that broad surveillance of the pathogen landscape in order to reduce the bias due to existing testing. So with that, I want to say thank you to all the study participants, as well as our collaborators at the BioHub and at the CHAMPS network. So I'm happy to take questions.